Okay, good morning everyone. It is 9 a.m. here in San Diego, and so we will start the webinar. Welcome to another webinar from MolSoft. My name is Andrew Ori and I will be moderating this webinar. Today's webinar is about a new product in MolSoft's suite of software called Scarab. It was developed in collaboration between MolSoft and the Structural Genomics Consortium in Oxford in the UK. Our speaker today is Brian Marsden, who is Head of Informatics Research at the SGC and helped lead the development of this software. Before I pass over to Brian, just wanted to let you know that you can ask questions using the chat dialog buttons in the GoToWebinar panel that you should see, or you can email me directly at andy@molsoft.com here. If time permits, the questions will be answered during the webinar, or we will get to them at the end. If you have a particularly long question, I can open your mic and you can speak directly to Brian. So now I'll pass you over to Brian. Thank you.
So while I wait for Andy to pass me over, let's just see, show my screen. I have to do a bit of clicking of buttons at my end. Hopefully now you can all see my presentation. Andy, can you see it okay?
Yes, we can. Yeah.
Okay, then I'll get started. So first of all, Andy, thank you very much for the invite to talk today about what we're doing at the SGC and how we've worked with MolSoft. What I want to do during this hour is a couple of things. First of all, I want to give a very brief introduction about what the SGC is about and what we've done and why we have been working with MolSoft to develop a platform that allows us to do good data management. I'm going to talk a little bit about what for us is good data management and how it helps us. And then I'm going to go into the actual product itself, which is called Scarab, and if time permits and if we don't have any technical glitches, I will be showing a few demos along the way to show you some flavors, at least basic flavors, of what the platform can do. It's very powerful. It's quite difficult to show everything we want to show in 1 hour, so I may gloss over some things. I will give opportunities for people to ask basic questions along the way, but if we can keep the more complex and deep questions towards the end, that'd be great. If there isn't time to answer those questions, then I'm obviously very happy to discuss the questions with you offline afterwards. So, if you let me go to the next slide. The SGC is a public-private partnership which was started in 2004, and we were originally based in the University of Oxford and the University of Toronto, and our purpose was to solve the structures of human proteins of medical relevance and put them into the public domain without any restriction. And we've been a world leader in this regard since 2004, but over the years we've branched out into other areas of primary research such as chemical biology, so the development of potent and selective chemical probes, particularly in the epigenetic protein families. Also focusing on developing renewable antibodies or binders, so recombinant antibodies using phage display technologies. And more recently we've been starting to use our chemical probes and our antibodies in the context of patient cell-derived assays with bespoke tissue platforms in particular areas such as oncology and inflammation. So this is quite a broad range of things that we do. We have been funded, as I say, since 2004. In the UK, we're funded by Wellcome Trust, which is the leading biomedical funding agency or funding charity in the UK. Large number of pharma companies. Currently, we have 8 pharma companies who fund us. And we have now sites not just in Oxford and University of Toronto, but also in Stockholm, in Campinas in Brazil, Chapel Hill at UNC, and in Frankfurt. And as I said up front, everything we do in the open. We pull out all our results, our reagents, and our know-how in the public domain straight away, and we never file for any patents. So as you can imagine from the sorts of science that we do, we have a very broad range of data types or things that we do or things that we make or data that we generate. And many of the workflows that we have are moderately high throughput. And so capturing this information and the pipeline progress is absolutely critical. And making that data available and interpretable is inevitably a challenge, particularly when you have an organization where we have people with skill sets all the way from molecular biology through to protein crystallography, through medicinal chemists, through to cellular biologists, who may not necessarily understand the data types and the interpretation of those data types. So back in 2004 when we started, we were fortunate fortunate enough to be able to impose that we were going to be a paperless organization. And we said that we would use a LIMS, or a laboratory information management system, and that everything we did would be captured electronically and make it possible to be able to share, and so on and so forth. And we've been working with MolSoft over the last 12 years to develop the capability to do this, and this has culminated in the Scarab platform, which I'll demonstrate to you today. I want to spend a bit of time just framing what a LIMS is and what I mean by the data management that I've been talking about so far. And for those of you in industry, none of this will be, or biotech, none of this will be a surprise to you, and maybe for some of you in academia, none of this will be a surprise to you. But I just want to frame this out and make it clear what I mean by some of the terms I'm going to use in the rest of this presentation. So LIMS is a laboratory information management system, and you can imagine the sorts of data you might want to capture in two ways. There might be what we call unstructured data. So this is data which isn't necessarily stuff that you would throw easily into a spreadsheet. So for example, an image, like this for example, a gel image, that you may annotate where the particular bands are, and you may annotate some quality or something like that. Or it may be a continuous readout, such as something from a purification column, for example. Or maybe it would be a cell image, where you could conceivably integrate over a particular color or a particular channel, but overall the actual information is somewhat unstructured. The structured data, as I sort of alluded to already, is the stuff that you would tend to put in a table like this. So this is where you might have, in this example, information about a particular purified protein, how it was purified, how much you made, how it was done, by whom, and by when. And a lot of this information associated with each of these records is what is called metadata, or data that describes what actually happened, or how an entity came to be. So when I use the words unstructured data, structured data, metadata, hopefully that frames what I mean by that. So why is LIMS a good idea? And this is the standard slide I show everybody, no matter whether they come from industry or biotech or academia. A lot of this is common sense. It allows you to organize your group's data, preserve that data inclusions, especially when someone leaves the group, share that information with your lab, and keep abreast of the developments even when you're traveling. It's also very useful for being able to collaborate as well. Being able to share what's happening in the lab with other people is good. These days, even in academia, intellectual property is a big deal, and being able to capture the information in a way that is suitable to be able to generate and justify any intellectual property that may come of it is critically important. And those of us in academia are painfully aware of the needs of funders these days, stipulating that we must have good data management capabilities to be able to deal with this. But as the cartoon on the right sort of tends to suggest, the key thing is you want to be able to capture what you did so that you can reproduce it, even if the person who is no longer there, well, he's no longer there. The person who did it is no longer there, so that what you do is reproducible. That's really critical. So these are the sorts of things that we were thinking about way back in 2004. The lab member leaves. 2 years later, where's the person's lab notebook? Or they can't read the writing, or they can't really work out what they did, or the person said they were keeping the lab notebook up to date, but it looks like they wrote very little, and that's really frustrating, and possible funding issues. Ability to be able to share internally has been a very big boon for us in terms of the LIMS. So these are the sorts of things that you tend to hear around the SGC. So someone tried a new methodology or a new tweak on a methodology and had some good luck, and then they'll say, well, can you look in my electronic lab notebook to see the gels? And that's often something we hear. Other things that we tend to do is often when we're sending protein crystals to the synchrotron to do X-ray diffraction trips. Some of these trips can be as long as just 24 hours, and the ability to be able to live blog what is happening within the LIMS platform, within the electronic lab notebook is really good. And overall, project management is simplified and it's much more efficient, and what we hope is that good science is promoted. And I think one of the good aspects of the SGC over the years is that we've developed a really good reputation of being able to produce reproducible data and reproducible protocols, and this has been a key aspect of that. So this is what the Oxford staff reception was like back in 2004. We expected them to say, well, we're used to writing up in a lab notebook, which is physically next to me. I don't really want people around me to see what I'm writing when I've not quite finished it, and I'd rather be in the lab writing up experiments doing experiments rather than writing many failures. But what we got was this. They said, well, it's actually quite easy to use. It makes my write-ups look professional, so it gets around the spider writing problem. Inevitably, people want to know where the spell checker is, and despite all this, people still don't want to write up failures. And of course, negative data is often just as useful as positive data. Sometimes there's just no hope. So we developed this mantra at the SGC that goes like this: if it's not in, it didn't happen. So in other words, if someone does an experiment and they come to a PI, then in general PIs have been conditioned, as it were, over the years to say, well, is it in your ELN, is it in Scarab, is it in the LIMS system? And if it's not, they have to go away and write it up. And this is the gatekeeper, if you like, the sleeping policeman that makes sure that the stuff actually gets in. And that's a really critical buy-in. You could have a wonderful LIMS platform that does everything you need to do, but if you don't have a way of forcing people to actually put it in and get them to see the benefits of doing that culturally, then it's a bit of a white elephant. So having said all that, those are the things that we wanted to do, the things that we have done, and now I'm going to talk a little bit about how we have developed Scarab in conjunction with ICM and some of the features that sort of cover some of the aspects that I have mentioned in some detail, and hopefully show you some demos that everything works okay, but let's see how it goes. So Scarab is actually part of the ICM family. So those of you who know ICM will not be surprised to hear that this is something that is integrated within the ICM platform itself. It's not a separate program, although it can be run in a standalone mode if you so desire. It features a built-in laboratory notebook, a very generic laboratory notebook, which I'll show you, and also data mining, extraction, and insertion capabilities, some of which are quite unique and quite powerful, as if there is time I will show you. Multi-user management in scientific labs and groups is a key aspect of it, and we believe it's a unified way of managing all our basic scientific data in one place and eliminating the data clutter, and also integrating with some of the key ICM product features, which hopefully I'll be able to show you. But as you can see from the diagram at the bottom, we have ICM, and Scarab is part of the ICM world, but it can interconnect or interrogate third-party databases such as Kemble or in-house databases, and I'll show you some examples of those as we go along. So first thing I'm going to do is to show you a few screenshots of what Scarab looks like, and then I'm going to try and flip into Scarab and show you some live examples of building some of the pages, as they're called, and also some of the structured queries and data adding, and hopefully everything works fine, but we'll see how it goes. So in Scarab there are two ways to get data in. There's a way to get the unstructured data in, that's the Electron Lab notebook capability, and there's a way to get data into the structured part of Scarab as well. Although, as I'll show you later, you can integrate the two things together, but for the purposes of the next few minutes, we're going to treat them separately. So here's an example of what we call a page, or a laboratory notebook page, and it's basically a set of sections that go from top to bottom, and these sections can be of different types, as I'll show you, but you can get the idea here that you've got text sections or image sections. You can put all sorts of images in, you can even annotate them, and so on and so forth. You sort of get the idea. If I flip to the next slide, you can get Excel sections in as well. That's a really important thing. I always forget to say that. So it will import Excel as well. This is what it looks like at the top of the user interface. Again, I'll show you this in a minute. But you can see that this section-based notebook has a number of different sections, so text, image, Excel. You can attach files, you can reference files, you can put in ICM sessions in there, and this result set thing, if there's time, I'll talk about later. But this is how we capture everything that we do. And so these pages at the SGC, we now have over 100,000, maybe 120,000 of them that have been written over the last 10 years, and it's a treasure trove of information as a consequence, because everything that is written in these pages becomes searchable because it's indexed appropriately. So, yep, here's some more examples of that. So now I'm going to try and flip over to the Scarab instance where we can do this. Andy, can I ask you a quick question?
Sure.
Can you see the right-hand side of this, or is it obscured?
Depend, do you mean that side? We can see. I just see the main. I can see the main ICM window. So.
Okay, good, right. I just wanted to make sure that everyone could see what was going on and it wasn't too small.
Yeah, nice.
So what I'm going to do is I'm going to create a new page. I hit this button here, and I have to apply projects to it. Now I'm logged in as a demo user right now, so there actually aren't very many projects in here, but you could imagine all the different sorts of projects as you define a project being in this list here, and you say, well, this experiment or this write-up is about this particular project. And then I can give it a title. So maybe I will say purification of CDK2 or human CDK2. Let's be a bit more specific. And I can say when it started. So it started today. And I can actually also add tags as well. So we have lots of tags in here, but we can build our own one. So like human CDK2, and this is useful for when you want to be able to search later on. But you can have as many tags as you want. So that's a useful way of doing that. At this point, there we go. And now I'm going to put a text section in. So I click on this, and I can double click and say, well, this is introduction. And then maybe in here I will write, and I want to purify CDK2, and maybe I want to kind of change the color scheme and so on and so forth. So there's all the usual sorts of things you'd expect. You can also copy and paste in from Word as well, and it will attempt to try and keep the color schemes and styles in a way that you would expect. You get the idea, that sort of thing. And then maybe I want another text session now, which would be, I don't know, materials and methods, and I would put my materials and methods in here. I'm not going to, for the sake of time, I'm not going to write anything in here, just to show you the idea. Now maybe I want to put it in an image, so I can click on image. Now the images come in here, so that's not in the right place. So I'm going to move it down by clicking on these icons, and then I can double click on it, and then I can import the image, which what you can see here is there are two versions of it. This is actually a very large image. Often images that come from gels, for example, come at very high resolution, and often there's not a particularly good reason for keeping it that high resolution unless you think you're going to need it for publication. So what Scarab does is it suggests a downsized or a scaled down version, and you can either tweak it yourself like this, or you can go with what it suggests, what I'm going to do, or you can keep the original. So that puts that image in like this. So now maybe I want to, well, I'm going to give it a title. So this is the protein gel. Ooh, I can't spell. Protein gel. And now I want to annotate this. So I'm going to right click, say edit image, and maybe I want to put an ellipse around this, a nice red ellipse, because that's actually the protein that I actually wanted off this. So I want to bring attention to that, and I can save that, and there it is. Other things I might want to do, maybe I want to put an Excel section in. I need to scroll down a bit, and so I can right click on this and I can import an Excel document. So if I import this Excel document from here, give it a moment to do its stuff, hopefully. The GoToMeeting thing is slowing things down a little bit, unfortunately. This is usually pretty instantaneous. The perils of doing a live presentation. Yeah, things have a little bit got stuck up. Well, what you will see, hopefully, eventually, is that the Excel section is shown in here. All the formatting is kept, but you can double-click on it, and then it'll open up in Excel. You can make changes, and then when you close Excel, it'll put it back into here. I don't know why that has all got hung up, but let me carry on with this instead. So other things that you can do with it is you can also import images using a mobile device. So you can look, there's an Android, and I think there might be an iPhone app that you can install on your mobile device, such as a phone or a tablet. And from this, you can then log into Scarab, and you can take a photo, as you can see here. And then when you put your image section in, you can then say Browse Mobile App Images. Click on the image, and the image will go in. So that's a very powerful feature as well. Other thing that we can do, which I'm not going to show you in detail now, is that if you're an ICM user and you're using ICM to look at protein structures or do computational chemistry of any sort, you can also import the ICB file, so the file that contains all the work that you've done in the ICM session, actually into a section here. So you can double click on it, and then it'll open up in ICM properly. So it's a really cool way of being able to capture what you've done as part of, say, a docking or a VLS run or homology modeling or whatever it is that you want to do. So at this point I'm going to pause and ask Andy if there are any burning questions at this point.
Yes, we've had a few questions. I'll start with, I'm paraphrasing some of these because they're written down, but so one question was how interactive are, for example, the plots that you showed? Are they linked to I guess it's asking if are they linked to a table or something like that? And also how interactive are the 3D sessions?
Right. So so for the plots, what you saw were images, and what we've imported are images. They're not actual links back to the raw data, not in this context. In the unstructured data component, which I'll show you in a minute, it is possible to actually have plots such as dose response curves, for example, although I will not be showing that today. So there are ways in which you can visualize the data that's stored in the back-end database. But for the unstructured pages, it's mostly putting in things like images and PDFs and Excel spreadsheets and stuff that's not quite so live from that perspective. In terms of the 3D visualization, it isn't actually live within the page itself. That would be really rather complex, and those of you who know ICM would understand that it wouldn't be particularly informative because you wouldn't have access to the rich controls that ICM has available. So instead, the decision was made that it shows a snapshot of the screen as you have it when you first open the ICB file, but then it opens up into ICM itself when you double click on it, so you can play with it in the correct environment. Andy, were there any other questions?
There was one question about indexing. How do you index? Is there like a Google search?
Great question. So let me see if I can pull up my other version of ICM here, of Scarab here. So this is a different. I'm logged in as myself now, so I've got to be careful what I show because I shouldn't show too much away. But what happens is that all the pages that are written are indexed as they are written, and it's not just the text itself, it's also Excel sections, PDFs where possible, and it's possible to then type things in here like CDK2, for example, and then it uses an elastic search capability in the background to be able to pull what you need. Or if it's not Elasticsearch, it's the MySQL, for those of you who know about the technology side of things, the MySQL text mining, which works pretty quickly and pretty cleanly. It isn't as fully featured as a Google search as we would like, although that is on the one of the next things that we want to do, and we're actively thinking about ways in which we can make it easier to not only do Google-like searches in the context of these pages, but also in the context of the structured stuff, which I'll show you in a minute. Anything else, Andy?
We had a question about different formats of data that you can import. One was, I guess, they're asking struct PDB and other chemical software, other chemical formats.
Yes. So the pages are really quite naive. They don't really understand things like SDF files or PDB files. Instead, you would tend to put in things like SDS, for example, into the structured part, and I'll show you that in a minute. Yeah, I think maybe the person who was asking this question was perhaps thinking about the idea of being able to be in ChemDraw and copy and paste into here.
Yeah.
That works if ChemDraw exports the actual image itself, but the actual embedding of the SDF in an integrated way doesn't work in that way yet.
Okay, great. We had a question, a specific question about can densitometry analysis be conducted on the imported images?
Scarab is good for capturing images, but not necessarily good for then doing downstream analysis on it in an integrated way. We've written Scarab in a way that we have tried to keep it as generic as possible and tried not to pigeonhole it into any particular workflow. Although, you know, obviously at the SGC we have a number of workflows that we use it for. We've always tried to be open-minded about it. As a consequence, hooks could be written from Scarab into an external program if that is supported by the external program. We have some examples in Scarab, for example, where we have written Python scripts that interact with the underlying database to be able to do some data analysis externally from Scarab, but can be triggered from within Scarab. But it wouldn't be Scarab that does it itself.
Okay, great. We have a couple of questions on authentication of particular users and administration. Is that something you're going to cover later, or did you want to answer?
I was going to cover it in a passing statement rather than actually showing it.
Okay.
So authentication at the moment is not enterprise ready, and what I mean by that is that right now authentication is done by the database backend, and which works for us fine. But we are expecting in the next few months to be able to cope with LDAP authentication. If you know what that is, that's a pretty standard way of enterprise level authentication, which is supported by Microsoft and Linux. This is actually something that we desperately need at the SGC, and something that we will be hoping to roll out in the next 6 months, certainly by Christmas. But it's a critical thing that we need.
Great. And the other question was, is it possible for the administrator to write a template for users to standardize their inputs?
That's a great question, and I can't believe I didn't think of putting a slide in this. Absolutely. Any page can be converted into a template, and then that template can be cloned if it's a standard operating procedure, for example, and the missing gaps filled in. And in fact, a lot of people do that here at the SGC, and I'm sorry I didn't explain that in more depth. But yes, that's a critical aspect of this.
Great. And we had a couple of questions about things, when you add things into Scarab, how interactive they are. But another one was, for example, when you import Microsoft Excel, is it fully functional within Scarab or only viewing?
No, only viewing, because well, okay, there are two reasons why we did it this way. The primary reason is, although it is possible to embed Excel into a Windows program using something called OLE, this is not supported under Linux because there is no Excel, and not supported under Mac because actually Microsoft didn't provide the capability for doing that. So as a consequence, we didn't go down that route. And the second reason is that we didn't want to constrain users' experiences to within Excel when there was a better environment elsewhere. So if you're going to do Excel-like stuff, you might as well do it in Excel. And then when you're happy with it, throw it into Scarab. And if you want to make changes, you double-click on it, it opens in Excel. It's an environment you're used to, and you know it's going to work. So those are the two reasons why we haven't implemented that way.
Okay, great. The other question was, actually, maybe we could leave this at, but for example, it's actually something maybe I should answer is this a module of ICM or is it standalone software? It's definitely
both.
Can be standalone, or you can import it into your current ICM.
Yeah, absolutely. I mean, I won't show it today, but you can run the normal ICM Pro, for example, session, and you can just connect to Scarab directly from within there, and you can use Scarab at the same time as using ICM Pro in the same program. You don't have to fire up another version of ICM Pro for that.
Great. And another question was, and you mentioned an app for Android and iPhone.
Yep.
So basically, is it? What's the functionality of that app at the moment? Is it?
So it is basically simply just to be able to. So the reason why we went down this route was we recognized that there were circumstances where people in the lab might want to take a photo of a setup or a photo of something and have an easy way to import that into the electronic lab notebook. So that's the main functionality that it has. We haven't gone down the route of porting Scarab to work in on a mobile platform for reasons that will become clearer when I start showing you the structured data component. But right now it's just for capturing images and finding an easy way to push it into your Scarab session.
Okay, great. Yeah, there's some questions we can answer at the end, I think. I don't know if you want to carry on. Yeah.
Yeah, I'll carry on then. Okay, so let's see, where are we up to? So the next part I want to talk about is the structured component, and this is a little bit more tricky to explain, so I'll try and go slowly through this. So the idea behind this is that you've got structured data that you want to capture, or you already have captured, in either a bespoke database or a public database like Campbell, and you want to be able to interact with that data either in the form of asking questions of it or modifying or adding data to it. And the way that we enable this is through a tree-like structure, which you can see on the left-hand side here. It might be easier actually if I move to the next one. So here, for example, is a a view of some of the main tables in version 21 of Kemble, for example. This slide's a little bit old, it's a newer version of Kemble since, but and what you can see here are some of the main components of Kemble, and what we can do is we can ask questions of Kemble by building what we call a query, or what other people might call a report, but in our jargon we call it a query. We're querying the data, and the way it works is we have a query builder which is on the right hand side here, and the query builder up here on the top right is split into two halves. In the top half we can drag and drop things from the tree in the left hand side to the right hand side top half to be able to constrain what we want, so to filter down what we want. And below that we can then also drag and drop other columns from the tree on the left hand side to be able to say what information that we want. And the really powerful thing about Scarab in this regard is that you can present the underlying database to your users in a way that makes sense to the users without them having to understand how the database is actually built in the back end. What I mean by this is that Scarab, when you point it towards the database, learns how all the tables are connected to each other and works out what are the best ways to join them when a question is asked across them. So, for example, in Kemble, we've sort of reordered some of these things here, and we have the user here has dragged and dropped things from different components, different parts of the database. They don't need to understand the underlying schema of the database, and then when they hit the run button, and here we're doing a substructure search, for example, a compound substructure search, and pulling information, then you get this results table here that can then be exported into Excel. And this is the real power of it. It is incredibly complex in that you can do almost anything you can think of, but that also gives you the ability to ask questions without having to have, for example, a new web page or a new web form having to build for every single question that you want to ask. So here's another example that we've pulled. This is where we're looking for something which is to do with the estrogen receptor from humans, and we'd like to see all the compounds in Kemble, for example, that are associated with that in some way or another. You get the idea. I'll show you a demo in a minute. It'll be clear to understand. This is something directly from the SGC schema, so it's important to understand that the schemas I'm going to show you today are not ones that necessarily come with Scarab. You can apply Scarab to any database that you already have that's an Oracle or a MySQL database, and connect them in Scarab transparently. It doesn't really care what sort of database you have. You just teach it what the columns are, what the tables are, how they join together, and the user doesn't need to understand all the IT components in the back end, as it were. So here's an example where we've pulled loads of structures and when they've been deposited, for example, and what the gene ID is, and we can export that into Excel as well. So I'm going to try and do a demo of this now and see if this thing actually restored itself or it got stuck. I'm just going to kill it. Now that died. So I'm going to do it in my other session, which is this one. So what I'm going to do now is I'm going to build a few simple queries, then build them up, and you'll start to see the sort of power that we've got. So we click on this button to start a query, and again, as I explained before, at the top half we're going to drag and drop information that's going to constrain or filter what we want, and in the bottom half we're going to put in columns that are the information we actually need. So what I'm going to do is I'm actually going to look in the SGC schema, so you're all going to have to promise that anything you see you don't repeat to anybody. I'm only half kidding, but I'll be careful what I show, it should be fine. So what I'm first going to do is I'm going to come up with a number of targets, or list all the targets to start with, I don't know, CDK, for example. And you can imagine, those of you who know kinases, the sorts of things that you expect to see. So I'm going to go into our target folder, and again I want to emphasize what you see here on the left hand side is a representation of the SGC database. This is not necessarily tied to Scarab, this is just how we use Scarab with this database. If you have a database that you already have in-house or one that you build, then you point Scarab to it and it represents that database in the way that you want. So don't think that what I'm showing you is the only thing that Scarab can do. It's very flexible. I'm just showing you in the context of what we do at the SGC. So what I always say to people when we're building queries is to think of what the query is as a sentence in English and break the sentence down. So what I want to do is I want to show all the targets that start with CDK. So I know that I want target, so I'm going to drag and drop this down into the bottom because that's what I want. I also know that the target has got to start with CDK, so that's a constraint, that's a filter. So I'm going to put that in the top half. And in here I can drop down, you can see all the sorts of things that you can do, but what I want is starts with, and then I'm going to put CDK. And you can see there's some auto-completion in there already, so already Scarab has asked the database what things are valid, but for the purposes of this very simple query, I'm going to just do that. Now there are other things I can do here. I can sort them, for example, and then I can run the query. And what it does then is it builds the SQL in the back end and sends it to the database and returns all the different values. And these are the CDK targets that we have at the SGC. And you see that's just one column. So this is very simple, a simple query within one table in the back end database, nothing fancy. But maybe what I would like to do is I would like to find all the targets that start with, I don't know, PIM, for example, which we sold a structure for and put out in the public domain. So again, that's a sentence. I'm going to break it down. I'm going to start a new query. I want to have a target that starts with PIM and has been deposited in the PDB. Now, again, this is an SGC specific schema. This is not tied to Scarab. I happen to know where this is. This is going to be in structured deposition, and I would like the PDB code for that. I can double click and it comes up. The date the deposition occurred. Maybe I would like to sort descending. Not on that one. On that one, that would make more sense. And you can see it puts placeholders in. These two constraints here are not active because they're not switched on. But I'm going to say that this one has to exist, otherwise we won't have it actually deposited. We have to actually have a PDB record in this table here. So I'm going to run this and hopefully it's going to behave and do what I expect it to do, but let's see. Oh, I'll try it again. Yeah. So here's all the structures we've deposited. They're all mostly of PIM1. What I can do now is I can export it to Excel. Let's see if GoToMeeting can cope with that. So what we always recommend to people is, you do your initial data mining within Scarab, and then when you've got what you need, you immediately put it into Excel because that's tends to be the environment that people are most used to, and they can manipulate what they want to do in that regard. Now, that doesn't look particularly magical, but I can tell you what was really cool about what just happened in that query was a very complex SQL query was built that jumped through approximately 16 tables. It's about 15 joins, all the way from target through our molecular biology, through our protein production, through our protein crystallography, crystallization, through the refinement of the structure, through the deposition. And our users have no idea this has happened, and they don't care, and they shouldn't care. And this is one of the most powerful features of Scarab in the unstructured area, in that I can build these ad hoc queries and not worry about how things are joined. So this is incredibly interesting. So other things we could do, I could now extend this query if I wanted to, so maybe I would like to, for example, know what the resolution is, and I happen to know where this is. This is going to be in here. This is comes in our data set. This is just how we capture things in SGC, and again, this is not Scarab specific, this is just how we capture our data, and Scarab is the layer over it. So I can put in the resolution in here, and I can drag and drop, and it will rerun the query for me. It's running a little bit slow, so then you can see the actual resolution. So you can see how you can build these queries up in a stepwise fashion. Now, these queries can be saved. So I can click on, I'm not going to do this now for the sake of time, but what happens is you can click on Save As, and then that query will then be saved in your folder up here, if it will open. GoToMeeting is a little bit slow on this. There we go. We have queries saved in here, or you can look in My Stuff, which has all my saved queries, which I can run in the future, and so on and so forth. So often what we ask people to do is to think about building template queries that they can share with others, or define what are the standard queries they're always going to want to run, so they don't have to rebuild them every time that we want to do that. I'm going to pause now because I've shown an awful lot in a very short period of time, and I'm sure there's lots of questions. Andy, are there any specific ones that we can answer right now? Hi, Andy.
Sorry, I need to open my mic. So yeah, one was related to what you just mentioned about the saving queries. Is it possible to share with your colleagues that query, or is
Absolutely. So you see this thing here says share queries. Well, what you can do is it depends how you set up your Scarab and what permissions you give it, but it's possible to have, to drag and drop your query into share queries or a subfolder within share queries, depending on who you want to share it with. That's eminently doable. Plus, it's also possible. I can go into users here, for example, and here's all the people in SGC. Within the permissions model that you have, you can then go in and you can start seeing all their different queries and all the pages that they've written as well. So all the unstructured stuff that I showed you earlier. So you can find stuff either by name, by date, by project, by searching. All these things are possible.
Okay. We had another question. I guess when you opened in Excel, are you able to point to a different data program such as a free spreadsheet software? I guess maybe Google Docs or something like that.
Yeah, that's a good question. Right now, you can right-click. You can export to a CSV file. You can export to an SDF file if there's a compound column involved. You can export to an XLSX file, and then those can be imported into other things like, for example, OpenOffice or Google Docs or Office 365 or whatever makes sense. We've only integrated Excel at this point because that's what works for us. But if there is a particular interest in implementing the capability to push to other platforms where that is technically possible, that's certainly something we would consider.
Okay. Great. Another one kind of similar is how can you see the relevant ELN page from a Scarab query?
Yeah, great question. I should have shown that as well. So let's see if I can find an example of that. So I'm going to extend this query now, and I'm going to put in, let's see if I can find what I'm looking for before I say it. I'm going to look up purification. So maybe I would like to know how the protein was purified for each of these instances of the deposited structures, for example. So I'm going to put I'm going to put purification ID in. I'm going to put it down here to save time. And then what you can see here is that this what is called ELN reference. This is a hangover from how we used to call it, but this is a direct link to the actual page itself. So I'm going to hope this works. We'll see how this goes. I'm going to run the query again. So you can see these are now hyperlinked, and I'm going to give this a go. I don't know how it'll work, but let's see what happens. So you can see I can say open that particular page, and here it is. So now I can drag and drop it, and this is Alex, who's one of our PIs, who was the one of the key, one of the people who really pushed the PIM1 program, and you can see all the information about how he actually solved that structure. Purify the protein in this case to be able to solve the structure. So that's how you would do it. So it's all integrated. Cool. Next question.
Question about actually there's some people from SGC. The question about it's pretty more an internal thing, but can they view these databases from other sites at SGC?
Yeah, and yes, and I will take that offline. I assume this is the guys from Brazil. I will definitely take this offline with you guys. There's something called Global Hive that I will discuss with you separately.
Okay, another one is, do you need to have MySQL or Oracle installed separately, or does Scarab come with MySQL installed?
Scarab is part of ICM. So it isn't something that comes with a database. It's something that connects to an existing database. So I will be brutally honest and frank, and I will say this is not something that you would install in on a PC somewhere and it would just magically work. This is something which is a bit more enterprisey and something for slightly larger organizations. If you have someone who knows a little bit of Linux or a little bit of systems admin capability and knows how to install packages, then it's not difficult to install MySQL on a Linux box, for example, which this thing that would then integrate with. And MolSoft have the expertise to be able to assist you with this, if you wanted to have a go at this. But this is not something that you just pick up and just run with. I should have said this up front, and I apologize if I misled you as a consequence. But you do need that back-end database, but it isn't as onerous as it sounds.
Great.
Andy, anything else?
Yeah, I think some of these questioners have strong backgrounds in the systems admin and stuff, so I think they'll be fun.
I figured, yeah. I figured.
So another question is, can you modify existing records?
Yeah, so that was going to be the next thing I was going to show.
How do you track changes? Sorry, and how do you track?
Yeah, I can talk about that in a minute. Are there any other questions, Andy, before I go to the editing bit?
Sure. This is, I guess, important. If the database is being accessed normally managed by a second tool, is there a danger of issues arising from overwriting data or formatting differences?
Yes, of course there is. There always are. But one of the things that we have tried to think about here is the issue of something called locking. So when some transaction, something is happening to the database locks that row or that table, depending on what sort of database it is and how it's set up, so that something else can't go and clobber on it. Now, Scarab is very careful to make sure it uses appropriate locking at the right times to do that. But let's say that you build a query like this, for example, and you run it, and you get a particular data set back, as I have now. If someone goes in now and modifies one of the PDB codes in here, this is not automatically going to be reflected in what I see here. This is a snapshot of what was in the database at the time I run that query. If I reran that query, so if I just clicked on run again, for example, then I would see that modification in there. When it comes down to modifications that occur when you're editing data yourself, again, Scarab is very careful about that, but there are always some scenarios that you can't legislate for, but we do think carefully about that. It is a serious problem, yeah.
Great. Okay, I think we can carry on then.
Shall we move on? Okay, right. So I've shown you the power of the sorts of things we can do with queries, but the key question is, of course, is how do we edit and how do we put data in? So what I'm going to do is I'm going to try to do, because this seems to completely seized up, I'm actually very quickly going to, if it will let me, start a new Scarab. And I'm going to log in as a demo user. So this is what it looks like. So here you see I have the server that I connect to, and I'm going to put the password in. So I'm not going to use the production server to show you the editing, because that would be crazy. I could probably get away with it, but you never know. So what we have here is a demo database that has very little in it, but I can do evil things to, and it won't be a problem. So I just wait for this thing to fire up. So what it was doing then, it was going to the database and saying, well, it's user X that's logged in. Tell me what things this user can do, what questions it can ask of the database. So that's what it was caching just then. So I've got this Scarab demo thing in here, if it will open up. Yep. And so in here we've got a basic table of genes, for example. So let's say I want to edit the data, so I can browse the data, edit the data, insert empty row. Let's just do edit data for now. So here's all my data. You can see we also have a card format down here. So if I click, it changes the card format. You can actually modify how this card format is shown, and that can be saved along with the query, but I'm not going to go into that today, so I'm going to switch that off. Now let's say I want to modify this, so I go into edit mode, and you can see there are certain columns which are colored up, and the columns which are colored up red, for example, are the ones that Scarab has found out from the database have to be filled in. So for those of you who know about the technical aspects of databases, these are the not null columns. Other columns may be grey, and those are the ones which the database that Scarab has detected may be auto-generated through, for example, some business logic or rule like a trigger, for example, but you can still edit it if you want. So let's say I want to add in a new target. So I'm going to say, I don't know, I'm going to make one up here. We're going to call this this target Scarab 1, just for the heck of it. And then here I might put me down as the owner. You can have drop downs in here. We don't have this in this particular instance, but in general what you would have for many of the columns is constrained vocabulary, or at least suggested vocabulary, so that you reduce the chances of having data in here which is not consistent and then hard to manage, or hard to query. So I'm going to put more stuff in here. So this is a test target. I'm going to put a dash in there, and the synonyms are, well, none. No synonyms, it is what it is, and so on and so forth. I'm not going to fill in the rest, but you get the idea. I can also copy and paste. So if I have this information in Excel, for example, or I have individual cells worth of data in Word or wherever, I can copy and paste in. So for example, if I copy from here, and put that in there, it will do the same. Other things that you can do, you can right-click, and if you have a numeric column, then you have things like filling down, and it'll also increment if necessary. So that's pretty cool too. When you're done, you hit the tick button, and it's in. Simple as that. So nothing goes into the database until you hit the tick button. So that's how we enter data. Now, if we have, I need to delete this data now because it's not real. So I click on delete. It's grayed out. It's naturally still in the database until I hit this button. It's in the database. If I decide that I'm about to do something stupid, I can hit the red X. You want to discard the changes, and it will put me back where it was. So that's how that works. So let's just see if this will behave. And see, it's gone. So that's how we edit data in general. It is not fancy. It is not particularly 100% user-friendly, but in the context of the way we work in our workflows, this works just fine. And the other thing I should say, which I think is in my next slide, we'll see if it shows up. Oh, I've gone back to the beginning. Just bear with me a second while I zip through. The next thing I should show you is it is possible, actually, if I can get my mouse to get in the right place, it is actually possible to embed these queries, this ability to show reports, but also to be able to put data in as a section into the structured pages. So this is useful in templates, for example, where you may be capturing, I don't know, the protein purification prep, where you've got to capture gel images, chromatography images, maybe mass spec, spectra, or whatever, and you also got to capture the metadata about, how much was made, or when or where, with which columns, and so on and so forth. So when you save the page, it saves the structured information into the back-end database as well as to the page itself. So you killed two birds with one stone. And this is a really important aspect of Scarab, the ability to be able to capture the structured and unstructured data at the same time. So I'm just going to move this out of the way because I'm having some difficulties here. Right, here we go. That's what I meant to do. Will it do it? Yes. So that's this. So other things I should show you is that MolSoft did a really good job of making it possible to import data from standard file formats, so for CSV, XML, SDF file formats. So you can import the file, it will detect what columns there are, and then it will populate them into Scarab, into the backend, either MySQL or Oracle database, depending on what you've defined as your default engine, and it will detect if it's an SDF file, if it's a particular compound, and if you have MolCart installed, the MolSoft MolCart chemical cartridge, then you can do things like substructure searching, such as this. So this is what was this that was entered? This was a little subset of Kemble that was entered. It then produced the tree for that little subset, and then we can build queries across the subset to get the results, which can then be exported either as SDF or into Excel. When there are compounds and you export to Excel, the images of the compounds are put into the Excel cells, so you do actually see that sort of information. So that is a very high-level view of some of the structured data aspects of it. Andy, I think time for more questions. Andy, you need to switch your microphone on.
Thank you for the reminder. Okay.
You're most welcome.
So we had one that you didn't answer from before. I'm not sure. Just double check. Yeah,
let's go for it, because we're basically at the end now. So
okay. So what is your experience with multi-institutional projects and institutional firewalls? For example, how my database is protected by a firewall. How can my collaborators use Scarab to access, import?
Yeah, it's really tricky. I mean, the problem is not Scarab, the problem is the firewall, right? If the institute is not going to allow you remote access to the database, then Scarab is not for you. Scarab is something that runs locally on your PC. It's not something that runs on the server per se. So all the magic happens on your desktop, either on Windows, Linux, or OS X. And it needs to have direct access to the underlying databases that you are integrating Scarab into. There is no other way around that. Apologies.
Okay.
Next question.
Great. Just one second. Yeah, there's a few little ones I can probably answer. They're more related to how to get the software and try it. So if you have any questions, please, any additional questions, please can you use the chat or email me, andy@molsoft.com. Alternatively, you can raise your hand and we can release your microphone and you can ask them over the GoToMeeting, whatever works best.
Yeah.
For you.
And this is the final slide, just saying some of the things I've already said. And this is basically what I've said. I mean, we have, although Scarab has been written with SGC in mind, we've tried really hard to not make it SGC specific and not specific to our particular workflows, in the sense that it should be possible to integrate it with any existing database, or if you want to build a database from scratch to capture your particular workflow, then the Scarab is quite useful for that sort of thing. And I think in industry where you may have legacy databases that are just sitting there and everyone's terrified of playing with, this is a really good way of being able to connect to that database and seamlessly, or at least transparently, be able to connect multiple databases together without actually modifying the databases underneath. So a classic example is Kemble, for example. We can hook Kemble directly into our SGC schema via Scarab. Scarab does all the magic in terms of being able to work out how to join across databases, even when the databases don't know that each of them exists, each other exists, and that's done without any modification of the underlying database itself, and I think that's a fairly unique concept.
Great.
And so overall, and the acknowledgements at the end, but yeah, open to questions.
Okay. I'd just like to say thank you very much, Brian. We really appreciate your time today. We have some questions, one related to the last question they asked, so about the firewall, etc. So can it be installed on a front-facing virtual machine that points to the database?
Yes. So here's the trick that we do at the SGC. So we have a Windows Server, Windows Server 2008 R2, which has Scarab on it, and obviously because it's inside our firewall, it has access to our databases. But because it's running on Windows Server 2008 R2, we can actually have Scarab run as a remote desktop app on our PCs wherever we are around the world. This is a nice feature that Microsoft have with Windows, and it means that there's an icon on the desktop which people double-click on, and it actually connects to that Windows Server, and it runs Scarab locally on the Windows Server, but projects the Windows actually onto your laptop or your PC wherever you are in the world. And this gets around a number of problems. First of all, it gets around the firewall issue, and secondly, it gets around the problem that talking natively to MySQL and Oracle databases is a very slow thing to do, particularly over a long-distance link. So this gets over those problems. So that's the trick, but that's not peculiar to Scarab or peculiar to anything that MolSoft do. That's just something that we've implemented to be able to get around this problem, and that works well. So we have people around the world who access our Scarab via that method.
Okay, thanks. We had a question regarding so you showed the query setup. So can you save a template for the query filters as well as the results? So both?
Absolutely.
Yeah.
Let me see if I can do it in this thing if it's not going to dial me. Let's see. Right. So let me build a query. Let me build a query in here. A really simple one. Let's say that I want to find the gene symbol. What I'm going to do first is I'm actually going to run something like this really quickly, just so I know what data there is, and I can show you. So it's a very small database, this. Okay, BRD2. Right, so what I can do is something called a parameterized query. So what I can do is I can say that I want the gene symbol to equal, and you see it's gone red because it's expecting something, expecting you to put something in. But if I save this query, so I save it as gene query, I do OK, and it says, I will say, oh yes, that. Yeah. I'll just wait for it to do its stuff. Okay, and then hopefully in my queries, there's the gene query thing there, and you see it's red. And if I double click on it, it doesn't actually. Normally, if I would double click on a green one that has all the parameters already in it, it would just run. But it can't run because I've not told it what the gene symbol should equal. So here I can put BRD2, and I can click go, and then it will run the query. The query is actually still there. It's in the back end here, but I left it blank. So that's a parameterized query. So it's a way of being able to, and there can be multiple parameters that are not filled in. So this is a way of doing template queries, if you like, and you can actually put in the information ad hoc and modify the query that way in an ad hoc manner. Hopefully that answers your question.
Great. Yep. Thank you. That's great. We had another question. Can Scarab be used for SAR?
Well, yes, absolutely. If you have, so the scenario I imagine that you're thinking that you have, if you allow me to interpret the question, is that you've got some compound data, some compounds with structures, and you've got some assay data, and you would like to be able to build an SAR table from that. Well, I would argue that ICM is the right tool for you to do that, and there are macros and capabilities within ICM, particularly ICM Pro and ICM Chemist, that will do a decomposition of your compounds into the different R groups, for example, and build things like SAR tables. But there's nothing to stop you from using Scarab as a means to be able to capture the compounds and the assay data. In fact, that's exactly what we do at the SGC. We have a big compound table that's all the compounds that we've ever tested, and we have multiple assay tables that capture that information. And then we can run queries within Scarab to say, well, show me all the compounds that have an affinity better than however many nanomolar or micromolar in a particular assay format, show the compounds, and then spit it out as an SDF file or put it into ICM for further analysis. Again, I want to really emphasize Scarab. We've written Scarab or built Scarab in an as agnostic way as possible in terms of workflow. So we've tried very hard to make sure that Scarab is not for a particular workflow to the exclusivity of all. We're trying to keep it open and generic because there are plenty of tools out there that already do many of these cool things, these questions are bringing up, like SAR exploring, and we just want to do the data management part and make it possible to be able to capture that information and export it or query it in a way that can then be put into the tool that makes sense for the actual question. Next one, Andy. Andy, I think you might have muted your mic.
Does Scarab come with embedded databases? I guess I can say.
So I'll repeat the answer to the question earlier, is that Scarab doesn't come with a database at all. But having said that, there are scripts that come with Scarab. So one thing let me backtrack a bit. One thing I didn't show you today, those of you who know about ICM will be very interested to see this. If I click on this, you'll see that everything you do in Scarab is actually an ICM command. And so everything I do is scriptable. And so what this means is that when you get Scarab, you get scripts that come with it, for example, that will take Kemble and they will build the Kemble database in the backend database and hook it up into Scarab, for example. And I think, I may be wrong, but I think there's also scripts for Drug Bank, for example. But everything is very scriptable and very flexible, and that's been one of our key design decisions up front, was that everything you do in Scarab has to be an ICM command. So those of you who are used to the ICM command line will find this is an absolute boon. You can do all sorts of really cool stuff.
Great. Yeah, and I'd also point out, usually when we send a trial of Scarab, we can send Kemble along with it. And
yeah, exactly.
So I should say that. Yeah, with this as well. Great. We had a question. Can you explain? They saw a option called lists and how to use it.
Yes. So I'm not going to demo that right now, but lists are a way of being able to create in your own space a list of objects like IDs, for example. So in this example here, it might be a list of gene IDs, and then you can drag and drop that list into this part of the query builder and constrain by a list of things. And that's quite useful if you have well, for example, a standard list of genes that you want to find assay data for example. And the list can be not just one-dimensional, it can be two-dimensional in principle as well. So you could have a pair or three or maybe four columns that will be constrained across. It's a very powerful feature, and I don't have time to show that today, but it's pretty cool.
Great. Yeah, I think we're coming to the end of the questions.
Good.
There's quite a few that, there's another one just coming. I can probably reply to some other ones offline. Just one just come in actually. Can other databases, i.e. non-chemistry drug databases, be connected? Yeah, I guess. Is it possible to be used for biology and chemistry departments, collaborative projects?
It depends upon what the database is and whether they'll open it up to you and what the schema of the database is as well. All Scarab can do is be pointed to a database and it will learn what it can from how that database has been written, what its schema is, how it's been designed. I should say that many databases are well designed, but there are many databases that are very poorly designed, and Scarab can only work with what it gets. And I would say that often third-party vendors' databases are not as clean and as clear as they could be, and therefore more work is required to be able to tweak Scarab's understanding of that database so that it's presented in a way that makes sense to the end scientist.
Great. Thank you, Brian. Just like to thank you again, Brian, and I just have one. People have asked how to get demo versions of this.
Yep.
And so hopefully you can see my screen.
Yep.
Yep. So basically you can email, as you've been doing, Andy at MolSoft.com or info@molsoft.com, and we can send you download instructions for Scarab, along with, as I mentioned, a Kemble example database. And I would.
Sorry, Andy, to interrupt. I would say I'm sure there are many technical questions. I mean, I've glossed over a very high level. There's a lot of clever stuff going on in the back end, and those of you who are technologically inclined will probably have a whole host of questions. Please don't hesitate to send them to Andy. Arman, who works at MolSoft, who is the brains behind most of this platform. I should have mentioned his name earlier. I apologize for not doing so. He will answer the questions as best he can, and of course I will help out too, and we'll endeavor to answer questions as quickly as possible. But no question is too simple or too complex. Just please bombard us with questions.
Great. Yeah, I might be forwarding you some actually for now. But great. So that basically ends the webinar. I just want to let you know about two that are coming up very soon. We're about to release version 3.85, and that includes some exciting new features. You can find out about what features are coming up in 3.85 if you go here. For example, there's a new 2D ligand interaction diagram, lots of new cool features in the 3D ligand editor for drug design, docking and such like, and also we support now this MMTF format, which allows you to read PDB files with millions of compounds in them. There's other features to be included. And finally, Ruben Abagayan, in a few weeks' time, I will send out the advert, but he'll be talking about MolScreen, which actually contains now more than 1,200 different high-quality fingerprint and pharmacophore models for a broad range of pharmacology and toxicology targets. So that will be another webinar coming very soon. Thank you very much for your time. As Brian mentioned, please go ahead and send us emails, and I'll forward them to Brian if needed, and we'll go from there. Thank you very much, Brian. Thank you.